Hello students, so today we are going to discuss about isolation of actinomycetes with VIVA questions. Okay, so another practical topic. So in masters you have this practical on actinomycetes. So a video is uploaded on my channel where a difference between actinomycete and fungi is explained, and another video where everything in detail with actinomycete, the structure, characteristics, then examples, then about different genus or um, difference in their spore arrangement, then economic importance, etc. is explained. Okay, so I will mention the link in description box. You can watch that video, and after getting your theory and basics cleared, you can watch this practical video. So actinomycetes are a diverse group of gram-positive bacteria. Okay, so they are gram-positive bacteria which are slow growers. Why? Because they have they have slow metabolic activity, and that's the reason they required minimum of 3 to 5 days to grow. Even some take around 7 days to completely grow and form a colony. So that depends on the species of actinomycetes. Then they are well known for production of different enzymes and antibiotics, and that's the reason they have many industrial applications. Then motility of spores is rare in case of actinomycetes, but if observed, then it is because of flagella. Then identification is done on the basis of morphology, colony morphology, then color of spores, production of diffusible pigments, production of melanin pigment, utilization of various carbon source, spore chain morphology, spore surface, means it has spikes or not, or the surface is smooth, rough, depends on that. Then biochemical test, and most important on the basis of cell wall analysis. So these are the criteria depending on which you identify your isolate. Then actinomycetes, they are a true bacteria. Okay, they are not fungus, but due to their colony morphology which resembles to fungus or fungi, many students get confused and they identify it as fungi. So they are not actually fungi, they are true bacteria, and they are placed in kingdom bacteria and a class actinobacteria. They are ubiquitous in nature, means they are present everywhere, but predominantly or commonly they are found in soil. They are also very common in marine habitat and are considered as a treasure house for secondary metabolites. Actinomycetes, they behave like decomposers. They carry degradation of organic components or compounds like chitin, then complex sugars, hemicellulose, etc. Then a good example from actinomycetes to remember is streptomyces is known to produce a metabolite known as geosmin. Okay, now which actually has a earthy odor. So when it comes in contact with water, it gives that distinct earthy smell. Okay, the first rain smell. The term is petrichor for that earthy smell which we observed on first rain. Then they are well known for source of antibiotics and bioactive molecules. Now in your Viva you may get questions on this topic, that is give example of say two antibiotics which are extracted from actinomycetes or any other bioactive molecules. So bioactive molecules means they can have antibacterial or antifungal or antiviral and antiparasitic activity. Okay, so that's why the term is bioactive. Hmm? So these are some examples, you can remember those. Then about classification. So classification is the domain of actinomycetes is bacteria, phylum Actinobacteria, class Actinobacteria, order Actinomycetes, and family Actinomycetaceae. So characteristics are like actinomycetes, they follow or they have following physiochemical properties. So this is actually a colony of streptomyces, and you can see how dull and dry it looks, and it is very tightly attached to the agar surface. Those white dots here, you can see it actually reflects the light, and it is the um, way to point out that the colony is very hard to pick, as it is tightly um, attached to your agar surface. Okay, so actually you need to dig in a part of colony to subculture. So that is the case in case of actinomycetes, but in case of fungi you can easily lift up the colony by using your forceps or loop, but not in case of actinomycetes. Okay, so that's the differential point which you should remember when you are studying with actinomycetes. So the cell of Actinomycetes, it has around 1 to 2 micrometer in diameter. They generally possesses a rod shape with a filamentous or branch structure. The filaments contains muramic acid. Most of the species are aerobic, while few are anaerobic to facultative aerobes. Then cell wall and internal structures are similar to bacteria. The cell wall of Actinomycetes contains mycolic acid. Okay, so you will get this in your VIVA questions. The growth of reproduction of Actinomycetes is slower than that of bacteria and fungi, and thus they are denoted as slow growers. Okay, but in case of fungi you will get growth complete or full colony growth is seen by 48 hours, but not in case of Actinomycetes. Okay, so Actinomycetes, they have around 60 to 78% of GC content, means they have high GC content compared to the others. Then Actinomycetes are most abundant in soil and marine habitat. The majority of species are usually non-motile, non-capsulated and non-acid fast. They show optimum growth at alkaline pH, whereas fungi, they need acidic pH. Colony is hard to pick from media plate, and you may observe Actinomycetes plate has a earthy odor or smell. When you are trying to isolate, you can just try to smell your plate from outside without opening it. Okay, so you may find little bit of that earthy odor or smell. So this is how your Actinomycetes plate should look when you are trying to isolate a particular isolate or culture. Okay, so you can see how powdery, dull and dry colony looks. Okay, and if you focus on a single colony, then you will see it has that cottony appearance, dry and dull, and it is very tightly attached to your surface, agar surface. Now about isolation. So CSA, that is casein starch agar, is media which is used for isolation of Actinomycetes. So this is the composition per 1 liter. And why to use CSA? So CSA includes complex substrates like starch, and it has high carbon to nitrogen ratio, and that is the um, main point, or say main characteristic of this particular media, which promotes the growth of Actinomycetes. But to inhibit the growth of fungi, we use griseofulvin, and for inhibition of bacteria we use ampicillin. So if you are not good with antibiotic calculations, then I have given here calculations for griseofulvin by assuming that you need to make casein starch agar the media volume of 200 ml. Okay, the media is to be made of 200 ml with griseofulvin of concentration of it as 100 micrograms per ml. Okay, so the role of griseofulvin is to inhibit fungi, and role of ampicillin is to inhibit bacteria. Okay, so now mode of action also you should remember, because if you are using this in your practicals, then examiner may ask you this question. Okay, so go through these calculations, and there are other two videos on antibiotic calculation available on my channel, where I have explained how to do it, so you can watch that again if still you have doubts. So other than CSA, that is casein starch agar, you can go for Actinomycetes isolation agar. okay, which is used for isolation and propagation of Actinomycetes from soil and water, and it contains sodium caseinate as nitrogen source. Other than these, glycerol yeast extract agar, that is GYEA, is also used, but it is a selective media to enrich uh, and for spore formation of antibiotic producing bacteria like Actinomycetes, Bacillus, and other groups. Okay, then few of the economic importances to mention here. So we know that actinomycetes, they produce several enzymes and which has wide range of applications in different industries. So lipase is used in detergent and pharmaceutical industries, cellulase is used in animal feed industry, catalase is used in detergent industry, amylase in food, textile and paper industry, and chitinase in biochemical industries. Okay, then Frankia is an example of actinomycetes which act as an symbiont that promotes root nodule formation and thereby nitrogen fixation. And even many of examples of actinomycetes, they play an essential role in bioremediation of organic compounds. Okay, so to just cover this point of economic importance here, I have mentioned these, but in another video of actinomycetes, different examples of actinomycetes which has economic importance is explained and mentioned. Okay, so watch that video. Now moving towards materials and methods. So materials, in that case we need media like sterile casein starch agar, then half strength casein starch agar for slide culture. Now the purpose of using half strength is, so half strength means you um, use half of your media components. Okay, so say you need 10 grams of starch, you will use 5 grams of starch in case of half strength casein starch agar media. Okay, then saline blanks, then media and reagents for biochemicals, garden soil or compost, sterile slide culture assembly, stains, glass spreader, alcohol, and microscope. Now about procedure. So say you have garden soil as your sample. So you will take 1 gram of sample, weigh it and add it to 10 ml of sterile saline in bumper tubes. Shake well and keep it undisturbed for 10 minutes. Okay, so this is your stock solution. From that, you will take 0.5 ml of supernatant and transfer it to 4.5 ml of sterile saline. Again, shake well and you use that to transfer or repeat the dilution. Okay, so you repeat 2 times more. That means you will have 3 dilution tubes. And from each dilution tube, you will transfer 0.1 ml of suspension and perform spread plate technique. Then in aseptic conditions, then you incubate your CSA plate at 30 degrees Celsius for 3 to 5 days. Okay, so generally you should observe or you observe growth in 5 days, but in case of some species of actinomycetes, they may take 7 days to show full growth. So after observing growth on CSA plate after 3 to 5 days of incubation, you mark different colonies. Okay, then you record colony characteristics and perform gram staining and inoculate for slide culture assembly, and you incubate your slide culture assembly. So between that time, you can refer Burgess manual for determinative bacteriology for biochemical test. Now after incubation of slide culture, again you record results for the structure of your actinomycetes and spore arrangement, and even you complete your biochemical test, so you can also report and record your observations for biochemical test. Now referring to Burgess manual of determinative bacteriology, you can identify your isolate till genus level. Okay, so this is a chart that I found useful. So it has different genus from Actinomycetes, okay, or Actinobacteria. For example, Nocardia, you can see here, then Mycobacterium, then Nocardioides, then Actinomedura, Thermomonospora, Streptomyces, Actinoplanes, etc. Okay, so you can see how their spores are different, the spore arrangement is different. Okay, and as Actinomycetes, they have substrate mycelia growing. That's the reason why they attach tightly to your agar surface. Okay, and that's the reason you need to dig in your agar. So this was all about isolation and identification. Now about Viva exam. So in Viva exam on this topic, you may get any of these questions. Okay, so first is what are actinomycetes, then how are they different from fungi, then they are gram positive or gram negative, then mention 2 3 economic importances with examples. Okay, then can they, oh sorry, why actinomycetes they grow slow. Okay, then name any examples of antibiotics which are obtained from actino and with their mode of action. You need to explain the mode of action as well. Then commercial applications of actinomycetes, then describe type of cell wall observed in actino, then why CSA is used for isolation. So even this I have covered in this video, answer is given. Have you heard of geosmin? What it is? So it is a secondary metabolite which is produced, right? And then why is half strength media used for slide culture? So the reason half strength media is used so that uh, initially the uh, half strength media will initiate the growth of your isolate, and as the nutrients deprive, it will enhance or it will promote sporulation of your culture. That's the reason we use half strength media. But in case of half strength media, we never cut down agar. Okay, agar will be same, the amount of agar will be same, just your other nutrients or other components we will cut those half. Then how will you differentiate between two cultures of actinomycetes? So firstly, depending on the colony morphology, and then second, by observing microscopic observation, okay, by observing slide culture, or by say uh, staining your actinomycetes for spore arrangement and the cell structure. Okay, so depending on these 2 3 characteristics, you can differentiate between your two cultures of actinomycetes. Okay, so I hope I have covered everything here, and I hope this video helps you out. So do like my videos, do share my videos with your friends, and do subscribe to my channel. Thank you.